So hello students and welcome back again to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the eukaryotic cell, that what is this eukaryotic cell and how does the process of DNA replication takes place in the case of a eukaryotic cell. So as you know that eukaryotic cell is completely different as compared to the prokaryotic cell. In my previous lecture, I have already explained it to you that in the case of prokaryotic cell, there was DNA polymerase 1, second and third which were being involved. We have also seen the structure of the DNA polymerase 1, which is also referred to as Kornberg polymerase. Kornberg polymerase. This is the Kornberg polymerase which is present. This is the Kornberg polymerase which is present, and this Kornberg polymerase is the one which is undergoing the DNA repair as well as gap filling activity. So there is the DNA repair activity as well as the gap filling activity that is going to take place. Okay, next point. In eukaryotic cell, nuclear DNA replication requires three different DNA polymerases. Those are DNA polymerase alpha, delta, and epsilon naught. So these are the three different DNA polymerases that are being involved. One is called as alpha, another one is called as delta, and the third one is referred to as epsilon naught. These are the three DNA polymerases that are present in the case of eukaryotes. Okay. Now, DNA polymerase gamma is a sole polymerase participating in mitochondrial DNA replication. Polymerase gamma is the one that is going to replicate, that is going to participate in the case of mitochondrial DNA replication, in which the mitochondria is going to replicate. Okay. Then there is the next enzyme which is referred to as the polymerase beta, which is present. Polymerase beta is present. Okay, so that is polymerase beta, nuclear polymerase beta, which is a small single polypeptide subunit which is present. It has diverse biological function. Okay, but biological role of the remaining polymerases are not known. DNA polymerase alpha is unusual because it has the ability to initiate a new DNA strand biosynthesis. So in the case of eukaryotes, the priming activity is being performed by DNA polymerase alpha. You look here, what is the activity of this alpha? The DNA polymerase alpha has the ability to provide the priming during the process of DNA replication. You all know that how the process of DNA replication is going to be initiated. It is always through the DNA polymerase alpha that will initiate the process of DNA replication. So through this alpha, we are going to initiate the process of DNA replication. Now, the next point, the DNA polymerase beta, nuclear polymerase beta, a small single subunit function in base excision repair. It is the one which is responsible for the base excision repair. Now, the main DNA polymerase. The main DNA polymerase are the DNA polymerase delta as well as the DNA polymerase epsilon. Okay. On the leading strand, the DNA is being biosynthesized by DNA polymerase delta. So, on the case of the leading strand, the DNA is being biosynthesized by DNA polymerase delta, whereas over the lagging strand, it is being biosynthesized by DNA polymerase epsilon naught. See this, DNA polymerase delta elongates the leading strand and polymerase epsilon naught may be involved in the lagging strand biosynthesis. So what we are going to see here is that firstly, during the process of DNA replication, the first DNA polymerase to bind is DNA polymerase alpha. Then after the binding of the DNA polymerase alpha, which is going to take place at the initiation complex, at the origin and synthesis of a short nucleotide chain is going to take place. 10 bases of RNA followed by 20 to 30 bases of DNA. It is being replaced by another DNA polymerase that is referred to as delta. Okay, and this delta is going to work over the leading strand. The replacement of the alpha DNA polymerase, replacement of the alpha DNA polymerase with delta DNA polymerase is referred to as polymerase switching. So what does polymerase switching refers to? The replacement of a polymerase with another polymerase is referred to as polymerase switching, in which one DNA polymerase is going to be replaced with another DNA polymerase. The mechanism of reaction is referred to as polymerase switching. Okay, now the DNA biosynthesis is going to proceed in a 3 dash to 5 dash exonuclease. In the 3 dash to with the help of DNA polymerase having 3 dash to 5 dash exonuclease activity, they are able to carry out highly processive DNA synthesis so that no wrong bases are being incorporated. DNA polymerase delta elongates the leading strand, and it is the gamma that is going to synthesize the lagging strand. The antibiotic that is being used, or a chemical agent that is referred to as F-E-dicoline, is a potential inhibitor of the mammalian nuclear DNA polymerase. So one of the most important drugs, or a chemical, a poison that could be used to halt the DNA polymerase, is referred to as F-E-dicoline. F-E-dicoline, which is a tetracyclic diterpenoid, is a potential inhibitor of the mammalian nuclear DNA polymerase. It is an inhibitor of the mammalian nuclear DNA polymerase, and which is being inhibited by the FeDeco line activity. Now, initiation. How does the process of initiation takes place? How does the initiation of DNA replication takes place? The initiation of DNA replication takes place by the melting of the DNA molecule. That means the two strands of the DNA molecule, when they separate, they are going to initiate the process of DNA replication. So when and how does the process of DNA replication starts? Just after the melting of the double stranded DNA molecule is going to take place, then the process of DNA replication is going to be initiated. DNA proteins initiate replication in E. coli at a particular point, which is referred to as the ORIC of the E. coli. This ORIC, which represents the origin of replication in the case of E. coli, consists of two different sequences. One is called as the 9-mer sequence, another one is referred to as the 13-mer sequence. I have already told you that binding of DNA A to the ORIC facilitates the initial strand separation, also referred to as melting. So it is the binding of DNA's A protein. You have already looked here that how the binding of DNA A was taking place. Okay, this is DNA A that would bind at a particular location at the 9-mer region here, and will cause the melting at the 13-mer region. Thus, strong region of a double-stranded DNA is being used in order to make a swivel and melt the double-stranded DNA at the 13-mer region. Then the DNA will melt at this particular region. Moving back to the topic, okay. Further melting of the two strands of the E. coli chromosome to generate unpaired template strand is mediated by DNA B protein. Once the initial melting has taken place, further melting is being taken place with the help of another protein, and that is called as DNA B protein, which is also a helicase in nature. Further melting takes place with the help of another protein that is referred to as DNA B protein, and this DNA B protein is also referred to as a DNA helicase. Okay. Next, after the DNA B protein is going to bind to it, the DNA B protein, which is a hexamer of identical subunit, is going to move. It will displace the two strands away from each other. Then the single strands of the DNA will be produced. You look here, one molecule of DNA B, a hexamer of identical subunit, clamps around each of the two single strands in the open complex between DNA A and ORIC. So DNA B is going to arise in between DNA A and the origin of replication. The in between the DNA A and ORIC, there would be a protein molecule that would be referred to as DNA B. Okay, this would be DNA B that would be present in between the DNA A and ORIC. Okay, the binding requires ATP and DNA C protein. Okay, helicase moves along a double-stranded DNA molecule utilizing the energy of ATP hydrolysis to separate the strands. So it is this enzyme helicase. Helicase is going to move. Helicase, both of these were referred to as helicase, whether it was DNA A and whether it was DNA B. Firstly, DNA A has caused the melting of the DNA molecule. On further on further action of the further action of helicase is being uptaken by DNA B, so that DNA B can carry out the unwinding process. This binding requires ATP and DNA C protein. Helicase moves along a double-stranded DNA molecule utilizing the energy of ATP hydrolysis. So helicase is going to move along the DNA double helix and is going to use the ATP hydrolysis energy to separate the two strands from away from each other. It would be using the ATP to separate the two strands away from each other. Once the two strands have been separated away from each other, the two strands are prevented from binding over further over each other with the help of another protein, and that is referred to as single strand DNA binding protein, also referred to as SSB protein. There could be another protein, and that protein is commonly referred to as SSB, single stranded DNA binding protein, which will bind to a single strand of the DNA molecule. It would be the one that would be binding to the single strand. DNA polymerase can only elongate existing primer strands of DNA or RNA molecules. Okay, so DNA polymerase can only elongate existing strands of the DNA or RNA molecule. The primer used DNA replication in both prokaryotes and eukaryotes are short RNA molecules which whose synthesis is catalyzed by the RNA polymerase primase. Okay, so the short nucleotide chains which are being provided are being extended by the DNA replicating enzyme that is referred to as DNA polymerase delta and DNA polymerase epsilon naught. What I am trying to tell you here, please listen very carefully. There are two enzymes which are present. One is delta, another one is epsilon naught. They are going to carry out the extension of the RNA molecule. The RNA molecule act as a template, act as a primer for the biosynthesis of the new strand. And RNA molecule is being biosynthesized with the help of a protein called as primase. Okay, see, DNA polymerase cannot elongate existing primer strands of the DNA. Primer used during DNA replication in both prokaryotes and eukaryotes are short RNA primers or RNA molecule whose synthesis is catalyzed with the help of an enzyme primase. See, primase is the enzyme that will be catalyzing the formation of the primer molecule. Okay, once the primase is going to work, it is going to form the primer molecule. After the bound primase synthesize short primer RNA complementary to the double stranded DNA molecule, they dissociated from the single stranded DNA template. It will get dissociated from the template molecule and will be completely separated. So this is referred to as the primase enzyme. Sometimes primase forms a complex with helicase. Primase may form a complex with helicase, and then it will lead to the formation of a complex, and that complex which would be produced is referred to as the primosome complex. Primosome complex. Okay, this complex is called as the primosome complex. What is a primosome? A primosome is a complex of primase plus helicase. When primase and helicase are being linked together, then it will lead to the formation of primosome complex. Okay. The next one is referred to as elongation. After the initiation has taken place, the next step that comes in the process of DNA replication is referred to as elongation. Now, during the elongation, step by step nucleotides are going to be biosynthesized or they are going to be added. So, step by step addition of nucleotides will be referred to as the biosynthesis pathway. Okay, so DNA polymerase catalyzes step by step addition of nucleotides, that is deoxyribonucleotide unit to a DNA chain. So once step by step deoxyribonucleotides are being added, the process is referred to as elongation. Now, in the case of elongation, I have already told you the nucleophilic acyl substitution was taking place. There is an incoming nucleotide triphosphate. This molecule indicate an incoming deoxyribonucleotide triphosphate. This incoming deoxyribonucleotide triphosphate will be attacked by the pre-existing nucleotide which is providing the 3 prime end of the DNA molecule. Okay, for example, this is a DNA molecule. There is another primer, there is another DNA molecule that is present, that is undergoing elongation. Then this end, which is referred to as the free 3 prime end, is actually this particular end. Okay, the 3 prime here, the hydroxy group that would be present here, the hydroxy group that would be present will be attacking a nucleotide triphosphate NTP molecule. It will be attacking the NTP molecule, nucleotide triphosphate molecule. And once the nucleotide triphosphate has been attacked, then the water is being eliminated. So you can look here that, see, this water is going to be eliminated. H2O, H and OH are there. Once H and OH are there, they are going to combine together and they would be relieved in the form of a water molecule. This is a water molecule that is going to be produced. This is minus H2O. This is a water molecule that is going to be released. And then there would be an oxyanion, O negative ion produced. This oxyanion will attack the positively charged phosphate molecule, because here it is negatively charged, here it is negatively charged, here it is also negatively charged, and this is positively charged. This is positively charged. Okay, this is positively charged. So it will attach the nucleotide triphosphate. And once it will attack the nucleotide triphosphate, it would be binding the nucleotide triphosphate with it. Okay. This is how the elongation is going to take place. This is the manner by which elongation actually occurs. So, elongation refers to a step by step addition of deoxyribonucleotide chains. The chain elongation reaction is catalyzed by the enzyme DNA polymerase, is a nucleophilic attack of the 3' hydroxy group of the primer on the innermost phosphate molecule. Okay, next. The primer on the innermost phosphate atom of the deoxyribonucleotide triphosphate, a phosphodiester bridge is being produced. There is another bridge that is going to be produced, and that is referred to as the phosphodiester bridge. So the next is a phosphodiester bridge is going to be produced with the commitment release of pyrophosphates. So there are pyrophosphates which are going to be released, and the bonds are being formed. Okay, so see this. Now a phosphodiester linkage will be produced here. This would be converted into a particular bond, and these two molecules will be released. So I'm just trying to make mark the molecules those will be released is these two phosphate. This particular phosphate molecule and the other phosphate molecule will be released. This is another phosphate molecule. Both of these phosphate molecules will be released in the form of pyroinorganic phosphate. This is pyro pi, and another one is pi. Okay, inorganic phosphate and inorganic phosphate, because the two inorganic phosphates are being joined with regard to each other, so this is referred to as pyroinorganic phosphate. When this pyroinorganic phosphate undergoes hydrolysis, water is being added, then what is going to happen? These two molecules are going to be broken down. This is one molecule, and there would be another molecule that would be produced, pi. So these are the two molecules that are going to be released. And this is a high energy molecule that is going to be released. यहाँ पर ये high energy molecule जो है ये release हो जाएगा. This is being released, making providing high energy for the reaction to take place. Providing the energy for the reaction, transforming it into a spontaneous reaction, making it into a spontaneous reaction reaction from a non-spontaneous one. with the expense of ATP molecule. Okay, so this is how the reaction is taking place. The subsequent hydrolysis of the pyrophosphate by pyrophosphatase, a ubiquitin enzyme, helps to drive the polymerization reaction in the forward direction. So you can see that the polymerization reaction is being moved in the forward direction. Next point. Elongation of the DNA chain proceed in the 5 prime to 3 prime direction. So the elongation chain is again going to proceed in the 5 prime to 3 prime direction, and the DNA is going to be biosynthesized. As each growing fork as each growing frog is going to move, it will lead to the formation of the leading strand that is continuously being biosynthesized. So, one strand which is going to be biosynthesized continuously will be referred to as the leading strand of the DNA molecule. Okay. And this leading strand will be biosynthesized in 5 prime to 3 prime direction. Growth of the leading strand proceed in the same direction as movement of the growing flock is going to take place. Synthesis of the lagging strand is more complicated because DNA polymerase can add nucleotide only to the 3' end of the primer molecules. So only the DNA polymerase is going to add the nucleotide only to the 3' end of the primer or growing DNA strand. Movement of the growing fork unreveals the template DNA for lagging strand synthesis in the 5 prime to 3 prime direction. So what I'm going to tell you here is that synthesis on the lagging strand is much more complicated. The synthesis of the lagging strand could only be understood with the help of the trombone model, as I have explained it to you in my previous lecture, where the new nucleotides were being added only when the strand was being displaced. Now, after the leading strand is being biosynthesized by the DNA polymerase, the strand which is being biosynthesized now is referred to as the lagging strand. We have already seen that how the lagging strand biosynthesis was taking place. Movement of the growing fork unreveals the template strand for lagging strand, which is going to move in 5 prime to 3 prime direction. This is the direction in which the lagging strand is going to be biosynthesized. After 1000 to 2000 number of nucleotides of the leading strand has been replicated, the first round of discontinuous strand synthesis on the lagging strand can begin. Okay. After the 1000 to 2000 number of nucleotides are being biosynthesized over the leading strand has been replicated, the first round of discontinuous strand synthesis on the lagging strand can begin. Now the next round of the lagging strand biosynthesis can take place. So DNA is being biosynthesized in short fragments, and those fragments are commonly referred to as the Okazaki fragments. So actually, DNA is being biosynthesized in small fragments in small patches, and the fragment that is being biosynthesized here is referred to as the Okazaki fragment. Okazaki fragments varies in size from 1000 to 2000 number of nucleotide in the case of bacteria, from 100 to 200 nucleotides in the case of the eukaryotic cell. So this is how Okazaki fragments differ. In the case of bacterial or eukaryotic replication, the replication is discontinuous. The strands are being synthesized discontinuously and they are referred to as the semi discontinuous mode of DNA replication. So here you can find there is a semi discontinuous mode of DNA replication that is going to take place. One strand which is being biosynthesized continuously is called as the leading strand. The other strand, which is biosynthesized in fragments, is referred to as the lagging strand of the DNA molecule. A strand of DNA that is biosynthesized continuously is the leading strand. Then there is another strand of the DNA molecule that is being biosynthesized in small fragments, and those fragments which are being biosynthesized is referred to as the lagging strand of the DNA molecule. This strand is commonly referred to as the lagging strand of the DNA molecule. Okay, so there is the lagging strand that is going to be biosynthesized. Okay, the proteins that are involved is DNA B, which is a helicase. Helicase combines with primase. Once the helicase and the primase combines with each other, then the helicase and primase combine. Then it will lead to the formation of the primosome complex. primosome complex. Primosome complex. There is a complex which is going to be produced, and this complex is called as the primosome complex. Primase and helicase combining together. Okay. The DNA strand which is being separated will be bound with another protein, and that protein to which they are going to be bounded is referred to as the SSB, single stranded DNA binding protein, that you can find here. These are the SSB proteins that are going to be bounded over the DNA molecules so as to prevent its over binding over itself. So these are all the SSB proteins that are there. Then after the SSB, the DNA polymerase is going to move in the two opposite direction. Now, because there is only a single DNA polymerase in the case of E. coli, as there is only a single, as there is only a single DNA polymerase, a single, as there is only a single DNA polymerase, polymerase as there is only a single DNA polymerase involved in E. coli. In E. coli, at both leading and lagging strand, at both leading and lagging strand, lagging strand. So, so the process is complicated. The process has been made complicated. The process has been made complicated and follows trombone model, follows trombone model of DNA replication, and so follows the trombone model of DNA replication. Trombone model of DNA replication, model of DNA replication. Now, how does trombone model of DNA replication takes place? That we have to see. I have already explained it to you in yesterday lecture, the animated lecture. Now, just to give you a view that this is how the DNA has been coiled. The DNA has been coiled in such a manner that, and both the strands of the DNA molecule, you can find that this is the 3' end. So there would be the other 5' end. Okay. There is a primer which is being provided. Primer is being provided, and then primer is being made to extend. Once you didn't look at this strand, now you just look here how primer is being provided. Primer is being provided with a free 5' with a free 3' OH. See this? This is the primer which is there. There would be the 5' end and the 3' end. This is the 3' end that has been extended. This would be the 5' end. This would be the 3' end. This is the 3' end of the primer that has been extended, and it has been extended in this particular direction. Okay, it is this 3' end of the DNA that has been extended. Okay, it is here the extension is going to take place. See this? This is how extension is going to take place. How? The 3' end is going to be extended by making of a new DNA molecule with the help of the DNA polymerase second, DNA polymerase third, another alpha subunit. You can look here. This is one subunit on the of the DNA polymerase which is working over the lagging strand, and then there is the other subunit which is working on the leading strand. So here you can find that the leading strand is going to be biosynthesized in a continuous manner. It would be continuously biosynthesized, and the lagging strand would be biosynthesized in small fragments. These fragments which are being produced are referred to as Okazaki fragments, and these Okazaki fragments will be uploaded. These Okazaki fragments, those which are being produced, those Okazaki fragments will be filled in with the help of the DNA polymerase enzyme. Okay, DNA polymerase second and DNA polymerase 1 are going to function, and then they are going to fill that Okazaki fragment. Okay, moving forward. Next point. See this, as each Okazaki fragments are being formed completely. If you have any doubt, you can send me the message. It's on. If any Okazaki, as each Okazaki fragments are being produced, the RNA primer of the previous fragment is removed by the 3' to 5' exonuclease activity of the DNA polymerase 1. So it is the DNA polymerase 1 exonuclease activity that is involved in the removal of the primer molecule. Okay, you can look here. RNA primer of the previous fragment is removed by the exonuclease activity of the DNA polymerase 1. DNA polymerase 1 performs the exonuclease activity, and it is the one that is able to remove the nucleotides. The enzyme also fills in the gap between the lagging strand, which then are being ligated with the help of DNA ligase. The last enzyme that is going to be functional is referred to as the ligase. Ligase is going to join the Okazaki fragments together with each other. Joining of the Okazaki fragments require an enzyme, and that is referred to as the enzyme ligase. In eukaryotes and archaea, bacteria, ATP is the energy source that is going to be utilized. You can look here, there is an ATP molecule. ATP is getting converted into an adenosine monophosphate molecule. Enzyme reacts with an ATP molecule. Once the enzyme reacts with an ATP molecule, then it will lead to the formation of an adenosine monophosphate, and nucleotide monophosphates are being going to be produced. You can look here, there is an AMP molecule. So just that, what I wanted to just speak to you is that either ATP is being utilized or NAD is going to be utilized. So both of these molecules are being required utilized, either one or the other molecule, as an energy source for the function of the DNA ligase enzyme. Ligase uses these as the AMP donor molecules. Last, once the DNA replication is being completed, see this, RNase H removes the primer. RNase, now the how the primer is being removed, it is the RNase H. There are two different mechanisms by which the primer can be removed. One is referred to as the RNase H mechanism. Another one is referred to as the fan endonuclease mechanism. Okay, fan endonuclease mechanism. Now RNase H removes all the nucleotides from the primer except the last three nucleotides. So the last nucleotide cannot be removed. So that to remove the last nucleotide, you require another special endonuclease, and that endonuclease which is present is referred to as fan 1 restriction fan 1 endonuclease. Fan 1 removes the last ribonucleotide. This is where the fan 1 is going to act, and this is the ribonucleotide that is going to be removed. Okay, it is here removed, which is going to be removed. Once it has been removed, then the gap is being filled, or the gap is being sealed with the help of DNA ligase enzyme. Gaps are sealed. Gaps are sealed with DNA ligase linking the two DNA molecules together with each other, and you have an intact double stranded DNA molecule. Some of the parameters to be compared between E. coli and human, you can find the E. coli DNA being 3.9 into 10 raised to power 6 base pair, 3 into 10 raised 3.2 into 10 raised to power 9 base pair for human DNA. Okay, total rate of replication is 1000 base pair per second. It is 100 base pair per second. Number of origin greatly varies. One, whereas in the case of eukaryote, there are various number of origin of replications which are present. Our to complete the genomic replication is 0.67. Here, the number of R's are 8 and so on. Termination. Now, how does the process of termination takes place? The termination of replication is also complicated in the case of prokaryotes, as well as follows different mechanism in the case of eukaryote. Now, what is the different mechanisms that are being used that we have to see? In the case of bacterial genome are replicated bidirectionally from a single point. There is a single point from which the replication of the genome is going to take place. So you can see that there is a point that is referred to as origin of replication. From this particular origin of replication, one replication fork is going to move in this direction, another replication fork is going to move in the opposite direction, that is in this direction, and both the replication fork are going to replicate the DNA. You can see here there are specific sequences which are referred to as the ter sequences, also referred to as the terminator sequences, which are present, as well as ter GFE B and C, GFB and C, and ter ADE sequences are present. These sequences allow the DNA molecules to pass through them in one orientation, while prevent the DNA replication fork to pass through in the opposite DNA replication fork. In the opposite DNA replication ter sequences. For example, supposingly, if this replication fork is going to move, this replication fork is going to move in this direction. These sequences will allow the replication fork to move in this direction, but will be halted at this particular location. At this particular location, the replication fork will be halted. And if the opposite is going to take place, if the replication fork is going to move from the opposite direction, I'm just taking another color for the sake of simplicity. If origin of replication is going to move, if the replication fork is going to move in this direction, then what is going to happen? Although the replication fork will move much more faster, you can see this is what I'm doing. The replication fork will be able to cross the ter sequences in one direction, but when they will reach the other ter sequences, the replication fork will be halted or stopped. So here the replication fork has been halted from this direction. This mechanism ensure that the termination of replication is going to take place in between the two ter sequences. Where is the termination going to take place? The termination is going to take place at this particular region, at this particular part. This is the part where the termination is going to take place. The ter A, D, E, ter C, B, F, and G are the sequences which are going to be present. So these are the terminator sequences that are going to be present. Okay. So these are the different ter sequences which are going to be present, and these ter sequences are going to differentiate each other. Ter sequences allow the replication fork to pass in one direction, but the other ter sequences which are present in opposite orientation will prevent the movement of the replication fork. The orientation of the ter sequence ensure that the process of termination takes place only in between the two ter sequences. Okay, see this. The orientation of ter sequences, the orientation of ter sequences, of ter sequences and bound TUS proteins, TUS proteins will ensure, will ensure that termination will occur, that termination will occur only in a particular region. Termination will occur only in a particular region. Only in a particular region. Particular region between the two ter sequences. Between two ter sequence. Okay, this is what is given here. When bound to a terminator sequence, a TUS proteins allow the replication fork to pass if the fork is moving in one direction, but blocks the replication fork if it is moving in the opposite direction around the genome. This directionality is set by the orientation of the TUS protein. Okay, the directionality is being set by the orientation of a protein, and that protein which is being produced here is referred to as the TUS protein. The directionality is being set with the orientation of the TUS protein on the DNA double helix. When approached from one direction, TUS blocks the passage of the DNA replication or the DNA B helicase, which is responsible for the progression of replication. But when approaching from the other direction, the DNA B is able to cross the TUS protein. When it is being approached from the opposite direction, the DNA B is able to cross the TUS protein effectively, so that the termination takes place only at a particular site. If you have any question in your mind, please do ask in the message form so that I can answer your queries. Any doubt, any doubt, any problem, please ask. So this completes the process of DNA replication. Now what we have to study is about the proofreading activity. Now what is this proofreading activity? How the proofreading activity is being performed? What do you mean by proofreading activity? Proofreading activity is a type of activity which ensures that effective replication is going to take place and the replication is going to be completed in an effective manner. Okay, now how this proofreading activity is going to be performed? The DNA replication is amazingly accurate. This is because of the proofreading activity of the DNA polymerases. The accuracy is necessary to keep the mutation load at a tolerable rate. The accuracy in the case of DNA replication is necessary to keep the mutation load at a tolerable level, especially in large genomes such as those of mammals. Okay, so here what we find is that the process is carried out in 3' to 5' exonuclease activity that is built into DNA polymerase. When the template primer DNA has a terminal mismatch, the 3' to 5' exonuclease activity of the DNA polymerase clips off the unpaired bases or bases. When an appropriate base pair terminus is produced, the 3' to 5' polymerase activity of the enzyme begins to resynthesize the nucleotides. I've already told you that how the nucleotides could be effectively biosynthesized, and if there is any wrong base that has been incorporated, the DNA polymerase itself has the ability of proofreading and it can remove that wrong base. Now, supposingly there is a DNA molecule that is present. I'm making a DNA molecule in front of you. There is a newly synthesized DNA molecule produced over this template DNA. And I'm taking the newly synthesized DNA molecule with green color. Okay, this is the parental and this is the newly synthesized DNA molecule. Now, supposingly this is a newly synthesized DNA molecule. This indicates new DNA, new dsDNA, whereas the one indicated with red color is referred to as the old dsDNA molecule. Okay, now the old and the new dsDNA molecules can be differentiated from each other because old has a specific methylation present over itself. This is the old DNA molecule. This old DNA molecule has specific methylations present over its structure. These are the methyl groups that are present over the old DNA strand. New DNA strand has methylation, but that is very, very less. Okay, it's very less, sometimes only in one or two strands. Then DNA polymerases differentiate between the new and the old strands on the basis of the pattern of methylation. DNA polymerases, DNA polymerase polymerase, especially the DNA polymerase 1, which is involved in the proofreading activity, is going to differentiate, will differentiate, will differentiate both the strands of the DNA, will differentiate both the strands of the DNA, both the strands of the dsDNA, of the dsDNA molecules, will differentiate both the strands of the dsDNA molecule and make changes only in the changes only in the daughter strand, only in the daughter strand, in the daughter strand, while the parental strand will remain constant, while the parental strand will remain as such. It will remain as such. There would be no changes that would be produced in the parental strand. The parental strand is going to remain as such, okay? And only the daughter strand, the green strand, is going to undergo modification. For example, if there is an adenine here, and this adenine is binding to guanine, now which is wrong? Should I replace adenine with cytosine, or should I replace guanine with thymine? I will have to replace guanine with thymine. Why? Because this is the parental strand. This is the new DNA molecule which is being biosynthesized. Proofreading only takes place at new DNA molecule. Proofreading only takes place at the new DNA molecules because it is partially methylated as compared to the parental DNA molecule, which is fully methylated. Okay. Please do attempt all these questions, that how Okazaki fragments are being produced. You can see the synthesis of Okazaki primer fragment is a key element for DNA replication. Each Okazaki fragment is initiated by a DNA by a RNA primer. How are these RNA primers being produced? The RNA primers are being made complementary to the DNA strands in the different direction by the enzyme that is referred to as primase. And the primase enzymes use several different types of nucleotide such as UTP, GTP, CTP, ATP, etc. After Okazaki fragments have been biosynthesized by the DNA polymerase third, the RNA primer is removed with the help of 5' to 3' exonuclease activity of the DNA polymerase first. So the RNA primer is going to be removed by the 5' to 3' exonuclease activity. Next, the gap that are being produced here are being filled in by the DNA polymerase 1 activity of the 3' end of the Okazaki fragments. Ultimately, the elongated Okazaki fragments are covalently joined together by the action of DNA ligase. Okay. Then there is a question to indicate that if the E. coli contain a defective ligase, then what is going to happen? The answer states that during replication of the DNA duplex, the leading strand is continuously being biosynthesized, lagging strand is being biosynthesized in fragments, and those fragments which are being produced are referred to as Okazaki fragments. Okay, which are then spliced together by means of DNA ligase. Mutants with defective DNA ligase produce DNA duplex in which one strand is going to be continuously biosynthesized while the other strand is going to remain fragmented. See, one strand will be biosynthesized continuously, that would be referred to as the leading strand, and another strand will be biosynthesized in fragments, that would be referred to as lagging strand. Consequently, when this duplex is denatured by the alkaline conditions, sedimentation results in one fraction containing the intact strand and one fraction containing the unspliced fragment. So one is referred to as the intact strand and another one is referred to as the unspliced fragment that is going to be produced. Now, the next is about telomerase replication and how does the telomerase act toward the end of the DNA molecule to complete the process of DNA replication. So, we're going to start this telomerase mode of DNA replication in our next class only, because I will be showing you the animation and then about this particular DNA replication. So, from my side, DNA replication is all over. We are going to start with a new animation tomorrow. This will be also completed, and then we will start with the mechanism of recombination. That how does recombination takes place? Please do solve all the questions related to DNA replication, and you can just ask me in the group. I'm making your group clean so that you can directly interact with me. Okay, so that's all for today. Good night. Bye bye.